Hey guys. So, my research was on elucidating the role of honeybees as biomonitors of environmental health research. I found this in the library under the insect section of the academic journal by MDPI. With the increasing rise of harmful pollutants into the environment that threaten our health, scientists are using honeybees as special detectors of these pollutants. With these increasing levels of harmful pollutants come increasing health problems for the environment and the people and the creatures and everything that resides in that environment. So these studies are important. And honeybees just happen to be the perfect study subjects because when they go out to collect food, they also collect pollutants from the air and the water and the soil, and all of this can be measured. The aim of this study is to monitor, regulate, and reduce toxic values in the environment through epidemiological research. Biomonitoring in particular is effective because it's cost efficient, it's cost effective, and it's efficient. There's a lot of reasons why honeybees are perfect biomonitors, but I'll just list a couple. They're ubiquitous, they're easily kept, and they're easily collected. And they conduct several foraging trips a day, sometimes up to 15, and they have a radius of about 7 kilometers. And one colony can gather up to 10 million microsamples of nectar and pollen in a day. They also gather honeydew, resin, and water. So since they're taking multiple samples from the surrounding environment, with all of this gathering, they tend to pick up different pollutants and contaminants. And when the bees return to the hive, the pollutants accumulate into the beehive products like wax and honey. The studies use these products to assess environmental contamination. To enable the evaluation of the honeybee sensitivity to these pollutants, the results are then compared with at least one site from a passive sampler or a different landscape type. So comparing contaminants between hives in heavily polluted areas such as industrial areas or urban areas with non-polluted areas like wildlife sanctuaries and in the actual wild. In these comparisons, it showed more than 82% of the studies used honeybees for biomonitors of heavy metal pollution. These studies were conducted over months and sometimes even years, and showed a steady increase of heavy metal pollution as time went on. To get more accuracy on the reliability of honeybees as biomonitors, scientists compared the values obtained using bees with the conventional physiochemical monitoring stations. And they discovered that the concentrations of both coincided with each other, but the honeybees revealed higher levels than the mobile stations did. The analysis of the whole honeybee allows the consideration of both internal and external contamination levels. Bees in different environments whose foraging practices differ from each other obviously are going to have different contamination levels. So the scientists monitoring 3 different colonies in the same sites to create a meaningful mean value. In contrast, they examined the variance between samples from hives in the same location to test for between effects and found no significant differences. The scientists also collected honey and honeycombs to assess the levels of pollutants. The contamination is believed to be caused by these pollutants from contaminated nectar and pollen or from the bee itself. They tested for 3 specific heavy metals: cadmium, chromium, and lead. And while testing in the more industrial areas, they found that significant difference in the cadmium content of the honey. It was not the case for the other metals. In addition to the hive samples, the researchers also took samples from the freshwater and from the soil and the environment surrounding the area. The analysis showed that there were more significant relationships between the water and the soil samples and the bees and pollen, N5, than with the honey, N1. In other studies, some areas in France were tested for lead levels in the honey, and the studies found that the lowest average mass fraction or concentrated hereafter were below the limit of detection in 30% of the cases and only detected in 10% of the cases. The scientists could not find any evidence of PA or polycyclic aromatic hydrocarbons transmitted from the honeybees to honey or the pollen. They found that the same results while testing levels of chromium and nickel, finding less seasonal variations and lower levels in honey. They concluded that the high heavy metal levels found in the honey may correspond with the foraging activity and the periods of continuous high pollution which allows the pollutants to accumulate in the honey. The results were that the honey itself was not a good biomonitor for heavy metal pollution. While it found honey to provide discrimination between polluted and non-polluted sites, in comparison with the bees and pollen, honey had the lowest concentration of the pollution. The results for the low usefulness of honey itself as a biomonitor lies in the honeybee's mechanisms to block pollutants as well as the physiochemical composition of the bee. Bees produce relatively clean honey because of a filter mechanism in their mouth. The physical barrier of the stylets of its mouth does not allow particles as large as or larger than 100 micrometers to be ingested. Another group of scientists studied the transference of toxic elements from bees to honey and calculating transfer coefficients. They found that the bees successfully prevented the transfer of two of the elements studied, cadmium and titanium. The article goes on for a lot more than that, and I encourage anybody to research into that if you're interested in this kind of stuff like I am. But the conclusion of the study found that using bees as biomonitors presented a promising avenue for continuous pollution monitoring. And using bees as biomonitors offers a more comprehensive and intricate perspective of environmental health and how it correlates with the animal and human health. And I will show you a couple things on this article. Sorry, I have to use my computer for this. I'm using my phone, but this is the heavy metals that they detected. And this is the polycyclic aromatic hydrocarbons. And then we have another table here that shows how many samples were taken from which hives, what kind of mattresses, what kind of pollutants they were testing for. And then these are all of the different studies that have been conducted on this subject. I hope you enjoyed this. See you soon.